Good day. Today, my group and I, which consists of Grecia, Flores, Ximena, Hernández, and Zharick Santoya, will be talking about transamination and deamination of amino acids. So, as mentioned, we will be diving into the essential biochemical process that allows us to recycle the building blocks of life, which is the amino acid metabolism. And this specifically focuses on two reactions, transamination and deamination. By the end of this session, you will be able to explain how amino acids are initially processed through transamination and subsequently prepared for excretion via deamination. You will also be able to identify the specific enzymes and cofactors and cellular locations, particularly the pivotal role of the liver mitochondria, and you will also be able to describe how these metabolic processes contribute to three key outcomes: energy production, the removal of nitrogen through the urea cycle, and overall nitrogen balance. And lastly, you will be able to connect the clinical relevance of these pathways to diagnostic indicators of liver function and the pathology of metabolic disorders such as hyperammonemia. So, as a brief introduction, we are looking at what happens to the amino acids after we consume, focusing on the nitrogen management. So we know that amino acids are incredibly versatile and that the body could use them for many things, which could be for generating energy, forming new glucose through gluconeogenesis, or even synthesizing fat for storage. And we know that these amino acids contain nitrogen, and the body must safely manage these components regardless of how the amino acid is used. So one way is to manage this nitrogen and free up the carbon skeleton for other uses is by two major pathways. And to put it simply, it's transamination, which is a transfer of the amino acid group from an amino acid to a keto acid, alpha keto acid, which prepares it for the nitrogen for later removal, and deamination, which is the final removal of the amino group. And this means that it releases it as a toxic ammonia that must immediately be expelled from the body, usually by urea. Okay, so in order to better understand this or in a more easier way, think about transamination as being the first step in breaking down amino acids. This basically is the first transfer or trade off. So you have an amino acid and you need to get rid of its nitrogen group. So what you do is that you, instead of throwing it away, you are going to pass it on to a keto acid, which is an amino acid without nitrogen. So you have an amino acid transferring its nitrogen group to a keto acid, and that will result in a new amino acid forming and a new keto acid. So it's a swap, a reversible swap. It's also important to note that this keto acid that you're transferring, this um, this nitrogen group to, it's an alpha keto acid. And this is how your body collects all of the scattered nitrogen into a centralized delivery place so that you could safely use it later. So, in order to make this trade happen, you need two things. Number one being an enzyme, and this enzyme is called an aminotransferase. And what this does is that it acts like a referee of sorts that manages and overlooks the swap. And two important enzymes or examples are the ALT and the AST. Secondly, you need a helper, and this enzyme needs a helper or coenzyme, which is called the PLP, otherwise known as the pyridoxal phosphate, which comes from your vitamin B6. It's also important to note that an aminotransferase can be specific to an amino acid or any particular member of a group of similar amino acids. And if you remember that I said that alanine aminotransferase and aspartate aminotransferase are two common types of aminotransferase. This is just the long name, and what I gave you at first was the short name, which is the ALT and the AST. So, where it happens and why it matters. If you put it simply, this happens everywhere. It could happen in the muscles, the kidney, and especially in the liver. And why do we need it? It's because it is essential in order for us to collect the nitrogen that needs to be removed from our body and to also build non essential amino acids that the body needs always. It happens in the mitochondria and cytoplasm of eukaryotic cells, and it requires synthesis of non-essential amino acids from essential amino acids obtained from dietary intake. Okay, so we look at an example, we would see that we have an amino acid, and this amino acid, the it transfers its amino group, to the alpha keto acid, and this is facilitated by the transaminase. And your products that you have is your new alpha keto acid, which the original amino acid is left behind as a carbon skeleton, and you then have a new amino acid, which the original alpha keto acid now carries the amino group. And what happens here is basically just the transfer of places. And if we use a real example, we have here alanine and alpha ketoglutarate. So alanine will be your starting amino acid, and your alpha ketoglutarate is the most important alpha keto acid in the body, which acts as a major nitrogen collector. Then we have your enzyme, which I mentioned, ALT or alanine transaminase, and this specific enzyme catalyzes the reaction. So here, the swap happens. Your alanine transfers its amino acid group to alpha ketoglutarate, and your products that you have is pyruvate. As I mentioned, the carbon skeleton is left behind by alanine, and this molecule can go directly into the process of making glucose. And then we have glutamate, and this new amino acid is created when alpha ketoglutarate accepts the amino group, and glutamate is the key molecule that carries collected nitrogen to the liver for the eventual removal via your urea cycle. So the process of transamination is basically to allow the body to have metabolic flexibility that allows it to move resources around. It plays an essential role in both the breakdown and the creation of amino acids. It is a irreversible process that allows the body to form non-essential amino acids that we need using the carbon skeletons and nitrogen from other existing amino acids, while also helping us for nitrogen collection as it acts as a funnel, channeling nitrogen from dozens of different amino acids into one central molecule such as glutamate. And this is the main nitrogen collector. If we look at the big picture or the overall importance, we could say that it helps it is important in the amino acid synthesis, building what we need and also breaking down what we don't need. This is a critical pathway that shuttles nitrogen safely from muscle to the liver, especially important during periods such as fasting.
Aspartate transamination is a reaction that involves the transfer of an amino group from aspartate to an alpha keto acid, most commonly known as the one that we have here, alpha ketoglutarate, which this will give us the products of glutamate and oxaloacetate. Regarding of the enzyme, in this case, we will have the help of the aspartate transaminase, AST, and also, even though it's not showing here in this image that I'm providing to you, the coenzyme pyridoxal phosphate, PLP, which is the active form of vitamin B6. Regarding the significance of this reaction, is a key pathway for both the breakdown and synthesis of amino acids. Also, that it will connect the amino acid metabolism to the citric acid cycle. As we have here, oxaloacetate, one of the products, is an intermediate in that cycle. And regarding the clinical relevance, the elevated levels of the AST enzyme in the blood can indicate a liver damage or other diseases affecting the organs where the enzyme is found, such as it could be the heart or the muscles. Alanine transamination is a reversible process in which the amino group from alanine is transferred to the alpha carbon of alpha ketoglutarate, giving us the products of pyruvate and glutamate. In this case, this reaction is key part of the amino acid metabolism and the glucose alanine cycle, requiring the help of the alanine aminotransferase enzyme, which will basically transport the nitrogen from the muscle to the liver. And because of that, it will require the help of the coenzyme pyridoxal phosphate, a form of vitamin B6. The reaction can run either direction. It will depend on the concentration of the reactants and products, which is influenced by the cell's metabolic state. Regarding in the liver, this reaction is crucial for gluconeogenesis because the alanine from muscle is converted to pyruvate, which the liver can use to make glucose. The resulting glutamate that we can see here can then be deaminated to safely dispose of the ammonium ions via the urea cycle. And this reaction will occur in the cytosol of cells, especially that will be abundant in the liver. Now let's continue about deamination. But what is deamination? Deamination will refer to the removal of the alpha amino group, resulting in the formation of corresponding keto acids and ammonia. This whole process will be in order to catalyze by specific enzymes such as deaminase and is linked to the metabolism of amino acids and carbohydrates. The initial step is basically the removal of two hydrogen atoms by the flavin coenzyme with formation of an unstable alpha amino acid intermediate. This intermediate undergoes decomposition by addition of water and forms the ammonium ion and the corresponding alpha keto acid, meaning that we will have a L-amino acid oxidase that occurs in the liver and kidney only. Now, what about the purpose of deamination? In their conversion to metabolic intermediates, the amino acids first undergo deamination. The primary goal of deamination is to excrete excess nitrogen as urea and then use or convert to glucose the remaining carbon skeleton. This deamination is a two-part process. The first step to deamination is usually a transamination catalyzed by an aminotransferase, in which the amino group of the amino acid is transferred to alpha ketoglutarate, which then yields a new alpha keto acid of the amino acid and glutamate. Key purposes of the deamination that we can find, for example, of energy production after the amino group is removed. The remaining carbon skeleton of the amino acid can be fed into metabolic pathways like the citric acid cycle to generate energy. Also, we will have the disposal of excess of protein. For example, when you consume more protein than your body needs, deamination will basically break down the extra amino acids as they cannot be stored. And also, we will have the removal of toxic ammonia because the amino group will remove from the amino acid is converted into ammonia, which is toxic to the body. So the liver then converts the ammonia into the less harmful compound urea through the urea cycle, which is then safely excreted in urine.
GDH deamination. This reaction is going to remove the alpha amino group from glutamate as free ammonia, and it's going to regenerate the alpha ketoglutarate, which can now be re-entering the TCA cycle. This reaction is going to provide us with NH3 or ammonia for the urea cycle, and it's going to link amino acid catabolism with the energy metabolism. On the other side, as you can see, I have an image, which this one is basically the representation of this reaction. Now we have serine deamination. This is going to be our non-oxidative deamination, and it's going to be serine to pyruvate catalyzed by serine deaminase. This is going to occur with hydroxyl amino acids or sulfur-containing amino acids. And the medical importance of this is the acid tolerance, because the deamination of serine is a mechanism that some bacteria, like uropathogenic E. coli, use to survive acidic environments. Talk about the relationship between transamination and deamination. So transamination, as we talked before, is a process where an amino group from an amino acid is transferred to an alpha keto acid, most commonly an alpha ketoglutarate, which is going to form a glutamate. This makes glutamate the central collector of amino acids in the body. Once the amino groups are concentrated on glutamate, the body can remove them through deamination, mainly oxidative deamination by glutamate dehydrogenase. This is going to release free ammonia, which would then enter the urea cycle for safe disposal. But because the amino group is first transferred to glutamate and then removed from glutamate, the two processes work together as a coordinating system known as transamination. This allowing nitrogen to be efficiently transferred, collected, and ultimately excreted. Now, as a conclusion, throughout amino acid metabolism, the body must manage both the carbon skeletons and the nitrogen component. Transamination plays a key role of redistributing amino groups and synthesizing non-essential amino acids. It also funnels nitrogen from amino acids like alanine and aspartate into glutamate. Deamination, on the other hand, is going to remove the amino group from glutamate so it can be converted into ammonia and processed in the urea cycle. The example of oxidative deamination of glutamate and non-oxidative deamination of serine show us how the body removes nitrogen through different mechanisms depending on the amino acid. Together, transamination and deamination ensure that nitrogen is going to be safely handled while the carbon skeletons enter pathways like the TCA cycle or gluconeogenesis. They finally are going to form an integrated system that is going to balance nitrogen metabolism and the energy production.